Welcome to Mex Academy. Today we will be looking at life sciences. Chapter 2. Meiosis 2.1 Definition of meiosis: it whereby a diploid cell or somatic cell undergoes two cell divisions, and divides to form four dissimilar haploid cells or sex cells. Somatic cell is any cell of a living organism other than the reproductive cells. A human diploid cell has 46 chromosomes in 23 pairs. Diploid cells have two sets of chromosomes, where each chromosome has a homologous partner. Haploid cells only have one set of chromosomes. Chromosomes in haploid cells have no homologous partners. Before meiosis begins, this is during interphase, DNA replication takes place. This results in two sets of chromosomes consisting of two identical chromatids joined together with a centromere. 2.2 The process of meiosis in animal cells. Meiosis is the type of cell division used to produce gametes or sex cells. The sex cells are sperm and egg cells. A cell undergoing meiosis will divide twice. The first division is meiosis 1 and the second is meiosis 2. In the first meiotic division, the number of cells is doubled, but the number of chromosomes is not. This results in half as many chromosomes per cell. In the second meiotic division, the number of chromosomes does not get reduced. 2.2.1 First meiotic division, prophase 1, chromosomes shorten and become visible as two chromatids joined by a centromere. Homologous pairs of chromosomes are now visible. The nuclear membrane and nucleolus disappear. The spindle starts to form. Chromatids from each homologous pair touch. The point where they touch is called a chiasma. DNA is crossed over or swapped at the chiasma. Crossing over is the exchange of chromosome segments between non-sister chromatids in meiosis. The spindle continues to form. Metaphase 1, the spindle extends across the whole cell. The homologous chromosomes line up along the equator of the spindle in their homologous pairs. One chromosome of each pair lies on either side of the equator. The centromere of each chromosome attaches to the spindle fibers. Anaphase 1, the spindle fibers shorten and pull each chromosome of each chromosome pair to opposite poles of the cell. Constriction. Telophase 1, the chromosomes reach the poles of the cell. Each pole has half the number of chromosomes present in the original cell. The cell membrane constricts and divides the cytoplasm in half to form two cells. This is the end of the first meiotic division. 2.2.2 Second meiotic division. Prophase 2, each cell formed during meiosis I now divides again. A spindle forms in each of the new cells. Metaphase 2, individual chromosomes line up at the equator of each cell, with the centromeres attached to the spindle fibers. Anaphase 2, the spindle fibers start to contract. The centromeres split and daughter chromosomes or chromatids are pulled to the opposite poles of each cell. Telophase 2, the daughter chromosomes or chromatids reach the poles and a new nucleus forms. The cell membrane of each cell constricts and the cytoplasm divides into two cells. Four haploid daughter cells are formed. Each daughter cell has half the number of chromosomes of the original cell. The daughter cells are genetically different from each other. An easy way to remember the events of meiosis is to use the word mnemonic IPMAT. Letter phase event, I and IPMAT stand for interphase, for in between, the part of the life cycle of the cell that is in between cell divisions. P and IPMAT stand for prophase, for preparation. The chromosomes prepare for meiosis by entangling and becoming clearly visible. Crossing over also takes place. M and IPMAT stand for metaphase, for middle. The chromosomes move to the middle or in the equator. A and IPMAT stand for anaphase, for apart. The chromosomes or chromatids move apart or move to the poles. T and IPMAT stand for telophase, T for terminal, the final phase of meiosis 1 or meiosis 2. 2.3 The significance of meiosis. Note that this can be asked in the essay question. There are two reasons why meiosis is important. 1. It reduces the number of chromosomes by half, in other words from diploid to haploid. This ensures that sex cells have half the number of chromosomes of other somatic cells so that when fertilization occurs the zygote formed has the correct number of chromosomes. It balances the doubling effect of fertilization. 2. Crossing over introduces genetic variation. Genetic variation results in offspring that are better adapted to a particular environment and ensures that they will have a better chance of survival. 2.4 Abnormal meiosis. Sometimes mistakes occur during the process of meiosis. This can happen in Anaphase 1 where the homologous chromosomes may not separate. Also called non-disjunction. It can also happen in Anaphase 2 when there is non-disjunction of the sister chromatids. If there is non-disjunction of chromosome pair 21 in humans, it leads to the formation of an abnormal gamete with an extra copy of chromosome 21. If there is fusion between a normal gamete and an abnormal gamete or with extra copy of chromosome 21, it leads to Down syndrome. 2.5 Differences between meiosis 1 and meiosis 2. In meiosis 1, the chromosomes arrange at the equator of the cell in homologous pairs. While in meiosis 2, chromosomes line up at the equator of the cell individually. In meiosis 1, whole chromosomes move to opposite poles of the cell. While in meiosis 2, daughter chromosomes or chromatids move to opposite poles of the cell. In meiosis 1, 2 cells form at the end of this division. While in meiosis 2, 4 cells are formed at the end of this division. The chromosome number is halved during meiosis I. The chromosome number remains the same during meiosis 2. In meiosis 1, crossing over takes place. While in meiosis 2, crossing over does not take place. If you have questions, please write them in the comment section below. Thank you for watching. Please don't forget to like, comment, share and to subscribe.